My name is Anireddy Reddy. I am a professor of biology at Colorado State University, Fort Collins, Colorado. Plants actually use multiple mechanisms to actually sense their environment. And in fact, my own research, we are interested particularly how plants sense their environment because stresses, the environment that they grow in, have actually tremendous impact on growth, eventually crop productivity. So in order to understand the effects of various stresses in the environment, for instance, temperature or drought, you need to understand how plants sense. So one of the things that plants use is they change the amount of cellular calcium in response to these various stresses, and that actually acts as a signal and then initiates a lot of biochemical reactions in the cell and then changes the entire gene expression. So reprograms the entire gene expression so that it now can cope with the surroundings. We use diverse tools, a number of tools. Ours is a mostly plant molecular biology lab, so we use molecular tools, we use biochemical tools, we use cell biological tools if you want to look at how a protein is moving in the cell, where it is localized. And also more recently, last 5-6 years, we are more and more using genomic tools where we are not looking at one single gene, but we are looking at all the genes, how are they behaving in a given environment. So we use a lot of high throughput sequencing using next generation sequencing. Now, in addition to second generation, more recently third generation sequencing is coming to place. So we actually use quite a bit of that last year using a third generation sequencing to look at how a plant cell is responding to its environment. So what we do is we basically take plants, seedlings that are growing under normal optimal condition, or you also take those same seedlings put under the adverse environmental conditions, for instance, lack of water, or you expose them to temperature. And then now you can now look at how the genes are behaving under these two conditions, and even as quickly as in 10 minutes, 15 minutes to over time of days. So not only just look at one gene, but every gene that is being made expressed, which means that's making RNA and protein, you can actually look at all of them, and then you can actually quantify digitally how much is changed and all of that. It's very instrumental now because as a graduate student, I was trained to look at, you spend all your life looking at one protein, one gene. But now, in order to understand in a holistic way how plants are responding to the environment, you cannot really just look at one. You have to look at the whole system. So at that time, the tools were not available because in 70s, only the sequencing came into existence in late 70s. So all you could do is sequence one gene. But now the technology is progressed so much, and also it's cost effective. So you can now look at the entire, globally, everything that is going on in terms of how many RNAs are made, how many proteins are made, what kind of proteins are made, and how are they changing in response to environment and all of that. And one other interesting thing that we work quite a bit is that not only you have a gene, it doesn't make just one RNA. It makes multiple RNAs. And this is one of the major findings after genome sequencing. Like when the people sequenced the human genome, they found out that we have only 20,000 genes. And then same as C. elegans, which has only 900 cells, it has only 20,000 genes. So then we think we are much more complex than worm or Drosophila. And then people started to look at why is that we have so few genes. And people, before the sequence, everybody thought, based on the complexity, that we should have at least 100,000 genes to 200,000 genes. And then they started to look at, it turns out that most of the 80% of our genes, they have these non-coding sequences in between. So when you make RNA, that RNA can be mixed and matched in different ways. So you have one gene, but you can make 5 RNAs, which means 5 different proteins. So it's not the number of genes that you have, how those genes are being used to make a protein. So it turns out that every gene that we have in our cell actually makes, if it has an intron, it actually makes 2 or more transcripts. So it wasn't known before the genome. And it turns out that plants also do the same thing. So most of, like 80% of the plant genes are also interrupted with these non-coding sequences. And then when they make RNA, they just don't make one RNA. They actually get mixed different parts of the RNA, they get spliced together, you make different RNAs. So our lab is actually interested in looking at how the stresses, the environment impacts that. In fact, we have shown that if you take a plant and is subject to a temperature stress, and as short as 30 minutes, you actually change which kind of RNA you make. Not only just whether it is making RNA, but which kind of RNA it is making. And we still don't understand all the significance of that, but quite a few genes, when you subject them to stress, they actually change this. We hypothesize that plants actually use that a means to quickly change what kind of proteins they make by changing how those messenger RNAs are spliced. And that's one of the hypotheses that we're working on. So since we can do globally, every gene that is expressed, we can look at it. So we can actually address those kind of questions because of the tools that are available now. Yeah, there are two things. When you think of agriculture and its impact on health, there are two things. One is whether we'll be able to produce enough food to feed the growing population, which means we need cultivars or the crops that are high yielding, because we already reached saturation in terms of the land that we can use. So only way we could increase the productivity to feed the growing population is by developing cultivars that can do well even under marginal conditions on changing environment, which means you basically need to understand how these plants grow under various conditions. Not only in various conditions, but it also has to be geographical location. So you may have a plant that does very well in place X, but if you take the same plant and put it in place Y, it may do terribly. So you have to do this, plants that are adapted to particular geographical location. So in terms of productivity, it may impact quite a bit of on health. But the other important thing is, even those in developing countries where there's quite a bit of food, but quality of the food is not there. The nutritionally poor quality. So we are used to eat a lot of food that is starch-driven but not balanced. So one way, all the tools that can allow you to do is to increase the nutritional quality of the food, so that your protein content, your antioxidants, all of these are vitamins. You don't have to take supplements. They're all enriched in the food. So that's all possible now with almost like a precision genomics. You could actually develop without much impact. As I was telling, with the genome editing tools, you could actually precisely modify stuff, a particular enzyme or particular biochemical pathway, and then you can actually change. In fact, golden rice is one of the best examples to just increase the vitamin A. So nutritionally, there is a lot of impact it may have because of these technologies. There are a number of labs that are actually working. In fact, one of my former postdoc, he actually has his own company. He's trying to produce vaccines in plants. So these are completely foreign to plants, but the technology is there now to introduce the piece of DNA or gene and then make proteins of interest. And then we are actually in one of the projects that was funded by Office of Naval Research. So they were interested in producing energetic materials that are important for Navy, but they currently mostly come from petroleum-based products. By producing those through petroleum-based products, you actually generate a lot of environmental pollution and all of that. So people recently identified genes that can actually make this in bacteria. So plants make some of those precursors, but they don't have all the enzymes to make the entire pathway. So what we have done is we took these bacterial genes and then combined that with plant genes, and we were able to produce novel chemicals that are never produced in the plant. So you can produce a lot of value-added compounds, like pharmaceutically important compounds, like vaccines, maybe antibodies, and all of that. The technology exists. So you can introduce entire pathways into plants. Like, for instance, people are now producing artemisium. The plants, only certain plants produce, but very low levels, but you can engineer the pathway. You can see which enzyme is critical for this production, and then you can engineer that. You can produce large quantities of that. Example like Taxol, which is used for cancer treatment. It's very expensive because plants produce very little. But a lot of labs are trying to understand the biochemistry of all the pathways. So what if you understand the fluxes, which thing goes where, and then you can actually shunt the stuff such a way that it now makes a lot more Taxol, or lot more artemisinin. It's all called metabolic engineering. You basically engineer this. You first understand the biochemistry, and once you understand what are the limitations in the pathway, and you try to address those, and then increase the productivity of these pharmaceutically important compounds. Sorghum is an important agricultural crop. It is the fifth most food crop. In fact, 500 million people actually depend in India and South Africa and Africa. A lot of people depend upon sorghum for food. It is also feed for animals. And then more recently, especially in the United States, it is considered as a e-crop, energy crop. The advantage of sorghum is that it can grow under marginal conditions. You don't have to have a lot of water because it's originated in Africa. So it is fairly heat and drought tolerant. So, and also it produces the most biomass. And also it does something, a different photosynthesis called C4 photosynthesis as compared to the rice, which does C3 photosynthesis. So it has a lot of attributes. Evolutionarily, because the way it was originated, it has a lot of things, but we still don't understand. Until recently, nobody paid attention because it's mostly poor man's crop. But now that US and other countries are interested in using that as a feedstock for biofuels, so they are now interested in understanding how this plant can grow under these harsh conditions. Still it can produce much more biomass and all of that, so that if you understand that, maybe you can utilize that even to transfer the technology or the information into other crops. So we are interested in, we have a project that is funded by Department of Energy to understand how this sorghum gene expression is impacted by the environment. So we are mostly interested in drought. So, and it's known for sure that most of the plants especially are very sensitive to the environment. If you take them from one condition to the next condition, it changes their whole kind of proteins that they make. But now we wanted to understand in sorghum, what are the proteins or the genes that make them? Sometimes they may not be protein coding genes. They may be RNAs, non-coding, but they may somehow regulate the cell metabolism in such a way that they become resistant. So there's a lot of emphasis on understanding what makes these sorghum plants more drought tolerant. And also there are some accessions that are very sensitive. So you can actually do this study in both cases and then see what is unique about drought tolerant. So and then now because of the next generation technology, so we can actually now not just look at one gene, but we are looking at entire genome globally. So that's one of the things that is ongoing in our lab now. It's to look at the impact of stresses on gene expression and how it is actually changing the DNA architecture. Because a lot of the expression depends upon how the DNA architecture is changed. So in fact, a lot of these environmental factors actually impact there is a question about nature and nurture. So the environment is basically an important part as to what genes are expressed. Those in turn actually have a phenotype on the plants.